All right, I'm told that we are live and on the air for our, I don't know what, fourth, fifth, sixth journal club. We have with us Steve Hill, and joining us for the first time, Joel Peterson, some fellow Marine I met at the RadFest, this past August. And we will be doing a paper that's, I guess, well, it's a paper that's talking about delivery methods of how to get things into cells, and particularly DNA. And they'll be looking at reprogramming factors, but pretty much this could be applicable to many, many ways of getting things into cells. Just before I go into the paper, just to give everybody kind of a landscape of what I mean about getting things into cells. So when you do genetic modifications, genetic engineering, or anything of that nature, or gene therapy for that matter, you need to take some sort of foreign body and usually some sort of polymer like DNA or a protein or RNA or something similar, and insert it past the cell membrane into a cell. And then once it gets into the cell, sometimes it needs to get into a particular location in the cell, like a cell organelle, or the nucleus. But once it gets inside the cell, if it's a nucleic acid, it then needs to be read by the cell's machinery, particularly polymerases and then later ribosomes, and then converted into something functional, right? And then the cell's functions are modified accordingly. So there's a couple of problems with getting this to work efficiently. One is the delivery method, right? So cells, typically cells don't like to take up just random foreign stuff from the environment. There are exceptions. Lots of bacteria love to do that. They'll take things up from the environment. But in general, mammalian cells are pretty poor at that. So people have developed a wide variety of techniques to basically sneak things past cell barriers and get them inside of cells. So just some of the couple typical techniques that are, available. You could use fairly crude approaches. With plant cells, there's something called biolistics, where you basically coat gold particles with DNA and blast them into the cell through the cell wall. For mammalian cells, you tend to use a little bit more finesse. You use usually modified viruses. So viruses can be basically a hollowed out protein shell, which is packaged with your DNA of interest. And viruses usually are specific for particular cell types, so they'll dock with markers that are found on cell surfaces, and they'll fuse and they'll dump their cargo. And there's other ways as well, such as electroporation. And this is what this paper will be looking at. A really kind of a more fine-tuned electroporation method. And electroporation is basically, like the name suggests, an electrical means to get polymers into, or molecules into, a cell. And it relies on basically a high voltage pulse, very rapid. And it induces the formation of these small nanopores in the cell membrane, basically breaking up the membrane temporarily and allowing things to essentially penetrate into the cell. So electroporation has been around for a long time. There are machines that are, basically a bank of capacitors, and you can adjust the pulse frequency. And you use electroporation on all sorts of cell types. You use it on bacteria as well as eukaryotic cells like mammalian cells. And this is what's referred to as bulk electroporation, where you essentially have a bunch of cells ex vivo, so you don't have cells inside of an organism, you have them outside. You put them into a special container called a cuvette that's got electrodes, and you basically pulse them with a high voltage electric charge, and some of the cells die, or a lot of the cells die. Those that don't take up the DNA, you grow them, and you hope that they're been transfected or electroporated with your DNA of interest. There are in vivo ways to do this, and I remember reading about them, where you essentially insert electrodes into, an animal, and you basically set up a positive and negative voltage field, and whatever's in between the DNA, essentially, it's pulled into the cells when the pores are essentially made. So this paper here that we'll be talking about. Let me just share my screen here with you. Share screen. Boom. There we go. Oops, wrong one. Okay. So the paper we're going to be discussing here, Topical Tissue Nano-transfection Mediates Non-Viral Stroma Reprogramming and Rescue. So what does that mean? So this is an electroporation technique that was developed by these guys. And it's called nano-transfection. So basically it's a really specialized electrode that has these micro or nano channels sculpted into it. And what they claim it does is it allows for much more efficient, highly focused electroporation into cells. So the electrode, it kind of reminds me the way they describe it. I don't know, those of you who've gone into a doctor and you've basically gotten, you know, a kind of an allergy shot, and it's got this little patch with a whole bunch of little needles, right? And it's like dermal, and you basically poke it, and it doesn't go all the way into your vasculature, but just into the dermis. So it's like a patch of little tiny needles. So it kind of reminds me of that. And I'm sure it's much, much smaller. So they call this TNT, I guess, for topical nanotransfection. And so it's non-viral, so not using viruses. And they're basically using it to transfect cells in the epidermis primarily. So basically the skin, right? And they're reprogramming them and seeing what happens in vivo after they essentially insert the cargo into these cells. So they're doing this with mice, okay? So let's take a look at some of these figures. Let's see, how do I move this out of the way? There we go. So figure one. So here in figure one, all the way in the upper left-hand corner, they show you kind of a schematic of their technique. So they have this little grid, this little electrical grid. Here's a 10 micron bar, and these little channels, these nano channels, where basically the substance is affixed. These channels are overlaid basically over the cell. So you have a 250 volt field. You have one of the poles go into kind of the dermis, the subcutaneous level, and then you have another pole up here, and then you basically have this as a pad over the surface. And they have to scrape the skin so you have contact, because the top layer of the epidermis is basically dead skin cells. So you need to make a nice contact. And the pulse is pretty fast. And basically this whole paper is about getting different substances that are coated onto this grid and electroporating them into localized regions of the mouse. So here's just a schematic of the nanochannels. And they're going to be using several reprogramming factors. This ASC1, BRN2, and MIT1, and this is gene expression relative to a control that they're using, which is BEP, which is bulk electroporation. So I guess they're using a more cruder approach, and to show that these levels, I guess these are relative numbers, are higher compared to the bulk electroporation technique. So the first thing they do is they use some sort of fluorescein labeled DNA, and they show that within like less than a second, you get, this is fluorescence over the area where they electroporated the mouse. I think that's a mouse feet, somewhere up on the, looks like kind of the thorax region. So they apply TNT, and you have this glowing patch of electroporated cells within like less than a second, I think it says. So 250 volts, 10 millisecond pulses, and 10 pulses, right? Yeah. So here's a cross-section of GFP expression from one of the pieces of DNA they transfected. Here's the epidermis and dermis, so you have expression. So they do a lot of other controls. So they look at gene expression in the epidermis. So these are the factors they've transfected. Gene expression relative to control in the dermis. And the significance of this is that realize that your electrode is up here on top of the epidermis, and they're detecting expression levels way down here. So they have to explain how some of this stuff is getting all the way through these cells down here. And one thing that they, I guess, speculate on, or they mention first, is that, that there are these EV vesicles, extracellular vesicles, that are known to basically ship cargo from cell to cell. And they basically, state that this is most likely how, this transfected DNA is getting past one cell to the other, is through these EVs that are being shed. And they do a gene expression relative to control, I believe, in these extracellular vesicles. If somebody can take a look in the materials and methods, I don't really recall how they isolated the EVs, but they mentioned that here they see, gene expression levels up in these extracellular vesicles. So, okay. So basically all of these figures here are just showing expression levels in the cells of various factors that they transfected. So this is a very topical way of getting things into a cell in a very localized manner. So where would the applications of this potentially be? Like, where would you potentially use this? And one thing, and I'm shifting over here to figure 2, one approach would be wound healing, right? Because if you have, let's say, wounds that are hard to heal, you need to promote growth of blood vessels or vasculature. So this is something that they look at. They basically pulse factors here, EFF. So these are, forgot what they stand for, but they're factors that are required for basically the transformation of epidermis to epithelial tissue, which is required for blood vessel growth. So they do the same experiment with a mouse, and these factors here, DAPI-PCAM and VWF, are basically factors that are specific for endothelial cells. And after this pulse, here's the control. This is a cross-section of the dermis. You have a lot more of these little speckles of red and green, which indicate potential vascularization happening. And this is just the percentage increase compared to the control. What they also do is they do these perfusion studies, high resolution laser speckle imaging, enhanced perfusion. So basically, they inject some sort of fluorescent dye. So you can see that, in an area where you don't have this electroporation, day 0, 1, 7, you don't have as much vascularization happening. You don't have as much perfusion of these, these fluorescent molecules that will basically track through blood vessels. Right, so this is just the perfusion ratio from control to transfected. And again, this is just a close-up of what's happening. This is some velocity measurements of some particles that they injected. They do a functional test also to show that there's this skin flap test where I guess I'm not too familiar with this surgical technique, but I guess what it entails is you probably isolate a region of the skin, and if you don't have blood vessel growth happening, you get tissue that starts to grow necrotic, or basically, slough off and die. So if you transfect these EFF factors, what they're showing here, I guess, this is the control region that wasn't electroporated, and this is also a control that hasn't been damaged. So at the site of damage here, I think you have greater necrosis happening for the control versus the EFF. Again, this is also this fluorescein test that was done to show vascularization, I believe. Yeah, laser speckle imaging increased blood flow to the flapped tissue treated with EFF TNT. So much more viable tissue and less necrotic tissue. So this technique could be applied for many conditions, where it's kind of localized damage has occurred and you need to get factors at that location. So figure 3 is basically a continuation of figure 2. So here they basically do a little bit more, dramatic approach, a surgical approach, where they do a femoral artery transection. So basically they cut off the blood supply to the leg. And when they do this electroporation, so here's EFF TNT. So with these growth factors that promote vascularization. Day 0, day 3, day 7, day 10, day 4. So this is the control. You could see that, I believe this is probably, this is the leg that's been operated on. You could see that the vascularization is not, the blood flow is cut off to this leg. And you can see quite clearly here that after, so day 3, you have this EFF-TNT method applied, the nano-transfection. And you could see, quite clearly see, a growth of a vascularization and transfer of fluids, fluorescent labeled molecules that are going throughout the leg. And they look at factors in muscle in the leg that are specific, PCAM, lectin, specific for angiogenesis, and these factors go up. So this technique, is pretty cool. We'd like to, I'd like to speculate to see where this could be, taken for humans. Obviously, wound healing, topical wound healing is one obvious approach, right? So if you have major damage to your skin surface or your muscles, or perhaps burns, this could be somehow, maybe this could be made into a sheet and applied to the surface, and to allow, for growth factors to be essentially transfected with a high frequency or, high efficiency to an area. They looked at relatively small areas, so I don't know how big this can possibly be applied to, but, it seems that, it's not really too much of a limit other than the size of the electrode. The only other thing that would limit it is at least the technique here does require skin abrasion. So, if you're going to be doing this, if there's already wounded, or if you have a burn, which is going to be abraded anyway, then you can probably use this. But, I'm thinking of, for people who are interested in aging, right? I don't know how applicable this would be as it now stands for, let's say, rejuvenating the dermis or the epidermis, right? If you want to put in factors, if you want to put in Yamanaka factors, for example, into an area, it might be an interesting experiment to do topically, right? If you have old skin and young skin, and use this approach and see if you can kind of optimize Yamanaka factor applications in one area versus another, and see if you can get a difference in skin rejuvenation and blood vessel rejuvenation in a localized area. And I'm not sure if there's any other ways we could modify this technique to be more useful kind of throughout the entire body, right? Because this is a localized electrode. Maybe there's some sort of nanoparticle approach in here that's possible. I don't know. I'll open this up to speculation. Anybody? Steve? Let me get back to, let me put my face back on. Yeah. So I should have been probably switching back and forth between my face and the picture. I'm told that that's the way to do it.
Yes, apparently people do like to see your face full screen.
Yeah. It's too bad we can't do it both, where we have the paper and the face, and switching back and forth.
Well, we can do, yeah. I mean, there are things we can experiment with if people want us to do that. If there's demand, we shall do it.
All right. Are there any questions? I didn't pause at any time for any questions.
Got nothing at the moment. I have just put an announcement in Discord as well. Nothing yet though, so I can only imagine they're completely enthralled by your excellent explanation there, or they don't understand it. It could be either. I might be in that camp myself.
Joel, anything? Pops?
No, just following you. Just following you. This is wild.
Okay.
Wild. I'm following.
So yeah. So I was reading the discussion, and usually this is where in the discussion section the authors kind of let their hair down a little bit, so to speak, and kind of speculate on kind of applications. There weren't too many far out applications speculated upon in this paper. I'm going to just throw out some half-baked ideas here because it's something I do. This idea literally came to me like while I was reading the paper and going over it a second time, just going down the stairs like 2 hours ago. I was like, oh, okay. You have an electrode, right? One negative, positive, and you have one inserted subcutaneously, one on the outside, and you have things going this way. Could this somehow be done with nanoparticles, right? Because people use all sorts of like nanoparticles for attacking tumors, right? And these particles are magnetic, right? So they can potentially be charged. Is there some way that you can do some sort of in vivo, in situ, internal electroporation, nano electroporation, like within organs without using these types of electrodes? I don't know. Maybe stick an electrode into somebody's mouth while perfusing them with nanoparticles.
It's all starting to sound like the plot of a sinister science fiction movie. But yeah, that's one of the limitations, isn't it? The deeper organs and tissues. I've seen a similar this technology used before.
Mm-hmm.
It's interesting, but it does have limitations, doesn't it?
Yeah. I could definitely see it used localized, for particular organs. Like if you don't need 100%, recovery, for example, like the pancreas, right? If you want to, regenerate, the beta islet cells to make insulin, I could definitely see this being, make a little incision and put that inside and transfect, areas within the pancreas. I could see something like that happening.
You could definitely use keyhole technique to get to most areas. The most obvious and problematic area, I would think, is the brain.
Mm-hmm.
That's, you don't really want to shove electrodes intracranially up your nose. It could be very difficult. But I mean, these things are to be overcome, aren't they?
Yeah, I mean, and certain other approaches too, like, the limitation of this thing, which is it's tiny, could also be an advantage in some situations. Like, for example, if you want to promote like regrowth of the retina in the eye, right? So you get some factors that are required for differentiation, and you have a little needle that goes back there, and you can just sort of reprogram areas that are defective, perhaps. I don't know.
Question?
I mean, there are other things. I'm gonna sound like a sort of a broken record here, but I'm going to mention my favourite subject again. I'm going to talk about senolytics. Of course, I am. It's me. I always talk about senolytics. Anybody would think I was something to do with a major mouse testing program, and we were testing them, right? But yeah, potentially we could use that to go back to what you said about skin. It would be fairly easy, I would think, to test it in a senolytic capacity potentially on the skin. A, because it's easily to access, and B, it is obviously, I would say, a relatively straightforward approach, isn't it? Senolytics. We know we know the sort of things that tip cells over that edge and cause them to destroy themselves. So, could we apply it in that way? I would think we could. Like things like the BCL2 family, which are known to make cells resist apoptosis. We could use it in that manner, certainly in skin cells, and it'd be quite easy and obvious whether it would work, wouldn't it?
Yeah, I'm wondering right now if one disadvantage again is that in these mouse studies they had to abrade the skin, right? If there's some way that you can have the electrodes functioning without having to abrade, the whole upper dermal layer, right? If there's some way you can make it permeable, I don't know, using DMSO, using something, that could permeabilize without damaging, the upper skin cell layer. Maybe a very mild, abrasion. Like if you go to like a dermatologist, they apply these like weak acid treatments, right? That sort of slough off your outer skin cells. So maybe you don't need like a very harsh abrasion, because people already do this to kind of to get rid of dead skin layers, right? So if you could do that,
exfoliating them, wouldn't
exfoliation,
it?
Yeah. How deep do you need to exfoliate? Because the other issue is how like they had to insert an electrode underneath, right? Because you had to maintain a polarity. So I'm not sure if there's a way to do that without having to subcutaneously, insert electrodes underneath all of the skin, right? If there's some way that you can sort of induce a voltage gradient from the inside versus the outside.
A little nanite, those little, if I'm following you guys correctly, how about if you even using a needle, like I work with my DO, we use needles to inject stuff. So if you throw a nanite type of environment that has its own little electrical impulse, you could send it into the cell, leave it there, maybe the cell repairs, and then program it a little bit later to do the electrical impulse from the inside.
Yeah, I was thinking about something even cruder. I was thinking of like if you want to get a more youthful face, right? You've got like an electrode like face mask, right? And then you have high voltage here that's like negative voltage, and then you need a positive polarity inside. So, since everything is in contact, could you just basically stick an electrode in your mouth and just bite onto it,
Exactly.
And then
I don't know, but I've got to say, an electrode mask sounds just very cool. I instantly want an electrode mask, even if it doesn't work. I just want one. So there you go,
And if we mold the electrode mask into different shapes, we can make you look like anybody you want. You could look like you could look like I don't know. Take your pick. George Clooney.
President.
George Clooney, Benny Hill. I don't know why I keep saying Benny Hill.
Oh God, no, not the president.
Because we need comedy in the world.
Shoot.
You could look like a youthful Donald Trump.
That's
I think I'd go for Nixon, slightly more popular.
A youthful Nixon. You could go for a youthful Nixon.
But yeah, poor old Nixon, eh? But everyone forgets that he started the war on cancer. Everyone forgets that.
Yeah.
Which is, obviously the war on cancer is progressing very well, isn't it? With immunotherapy nowadays.
Yeah. He also led to the détente with China.
He did,
right?
Yeah.
Yeah, he was one of the. I think he was the first U.S. president to visit China, wasn't he?
I think he was actually.
I believe so. Yes, I think so.
But yeah, I mean, to go back to cancer as well. I mean, obviously immunotherapy is coming along as well. But could we even potentially use this technique in the war on cancer? I mean, if we can potentially use it for senolytics, which tips the cell to destroy itself, could we perhaps use it for cancer treatment? Could we attach, the electrode to tumors and activate very similar and related pathways to senolytics that also stop the cell resisting apoptosis? could we apply it to cancer? You know, could it be used for the war on cancer? I'm thinking things like melanoma in particular.
Yeah. It could also be used more subtly. Like you could maybe cause the cancer cells to express factors that attract, you know, natural killer cells or something like that, right? So you could alter the tumor microenvironment to become, you know, less conducive for the cancer cells to grow and attract immune cells. That's possible.
Or track them, maybe track the cells and see what they do.
Or track them. You could put in any kind of factor, basically.
Well, you know that a lot of cancers actually disguise themselves. They go into a stealth mode that stops macrophages and T cells spotting them. I wrote about this a while ago on Leaf, but they found that they can actually they can basically use this. They express this "do not eat me" signal. I suppose that's what I want to say.
Yeah,
because it's the most easy to understand thing. I can't remember the exact pathway it uses, but it literally sends out a signal that healthy cells in the body also use to identify self. Because otherwise, obviously, you don't want your immune system to start attacking itself. So healthy cells send out "do not eat" signals. So cancer also is clever because it also hijacks this pathway as well and sends out the same signals. So my question is, could we potentially use it in tumors, which obviously are in one place and are growing? Could we use it to turn off the "do not eat me" signals, so then that they would be recognized by the rest of the immune system, and then the macrophages come in and go, oh, nom, nom. You know, because macrophages do that. You know, they're like basically like Cookie Monster out of Sesame Street. But that's what a macrophage is. I mean, it comes in and goes, oh, nom, 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 nom.
Yep. And
I've actually used that. Someone said, well, what's a macrophage? And I said, well, you know, Cookie Monster basically is that, which it is, kind of. But could we use it to switch off the "do not eat me"?
Maybe.
Yeah, you know, has anyone thought of that? See, you know, I think there could be good application for cancer there.
I'm still, my brain is still worrying, thinking about ways to kind of modify this approach in some sort of way, nanoparticle wise, for a whole body type of reprogramming. Because delivery is always the problem. I don't know if this is applicable. Maybe somebody can think about it a little more. Oh, did we just lose Joel?
Oh, hopefully he'll rejoin.
Yeah. Oh, he's back.
There we go. Oh, we've also got a comment from a viewer. Also, apparently Keith Comedo, I don't know who he is, the viewer, and he said, "Oh, that was a sick Nixon burn," and apparently he would also like an electrode mask as well. That sounds cool.
Keith would like an electrode mask. He would like
an electrode
in the shape of Nixon.
Oh, I don't. He didn't specify whether he said Nixon, but he thought it was funny.
I just.
But you know, I was actually just trying to tie. Basically, with my Nixon comment, I was just trying to tie it all into the war on cancer. Really, it was my very not subtle way of sort of easing the conversation towards cancer, because I'm always talking about that.
Yeah, and then I just started to veer it off more and more into the geopolitical stuff, and then you got me back on track.
It could be worse. You could be talking about being an energy being or a sentient nebula.
Yeah, I'll leave that to other organizations.
Keith wanted to be the 2001 space baby or something.
I'll leave that to Kurzweil.
Yeah, I think I'd rather be Hal, actually. I can't let you do that, Oliver.
Oh my God. Okay. Is there anything coming through the pipeline question wise? Because we could be riffing on this all day.
So let's look at the moment. So how do you think it compares to traditional methods of gene therapy? How do you think it ranks up?
Well, if it's I haven't compared, I haven't looked at and compared the efficiency to other methods in vitro, but one thing I'd be curious about seeing is, and I'm sure they have the data there, I just haven't gone through the supplemental, is how does it compare to viral delivery and other delivery methods in vitro, right? Because that would be pretty simple. You just have a plate with an electrode, right, and coated, and what would be the efficiency there? So that would seem to be, if that is at least as efficient as using modified viruses, then it could potentially be way cheaper and way easier to modify cells ex vivo before reintroducing them into cells without even, and maybe even avoiding certain BSL2 vectors like lentiviruses and that kind of thing. If it's an ex vivo approach, if it's inside of a dish. So, and of course, we also have to look at different cell types, right? Because they only looked at the epidermis here, like how. If we look at cells in vivo or in vitro, can we take a look at cells in vitro, like different cell types, and see what the efficiency is comparing them all? So maybe this is a broadly applicable approach that could be marketed for gene therapy, where you're taking cells out of a person and modifying them outside. And the less vectors you use, the less viruses and whatnot, the less the less issues there are with handling and other types of things. So I'd be interested in seeing where it goes in that direction. But as far as in the organism itself, the paper doesn't go deeper than the epidermis here. So obviously, you still have a need for viral delivery methods, right? One advantage of viral delivery methods also is that they travel through the vasculature, so they can get into all the nooks and crannies if you're going to be perfusing, for example, a liver or something like that, versus this type of technique. That's why I keep thinking, is there some sort of way that this could be done using nanoparticles that are charged, right? And you have an external charge somehow applied temporarily, where you basically give the organism, basically can make the organism give it a high voltage pulse without frying it, and having the vesicles deliver their cargo once they've kind of circulated around. I mean, one issue with viral delivery methods and other, well, not really viral delivery methods, but methods that are not very cell specific, is if you inject them into a local area, they're going to start transfecting cells, right? So they won't travel very far before, if you want to get them to go everywhere, they're going to transfect a localized area. So I'm wondering if this could be an approach where it could be used to perfuse an entire organism, and then once these particles are circulating throughout the entire organism, you then apply a pulse, and everything gets transfected at once, because the particles are evenly distributed. How that would be done, I don't know. I'm just throwing that out there as a completely half-baked idea, because why not?
Well, no, it's not necessarily half-baked, is it? I mean, it's an interesting idea. The key would be how to get those particles distributed evenly throughout the target tissue.
Yeah, I mean, you'd assume they'd circulate around enough, but then the thing would be, how would I get the voltage to? How would I get the particles? How would I make a polarity difference between the particles and the rest of the tissues? Right? Is there some sort of way to do it using MRI? I don't know. I mean, I'm not a physicist, so I don't know if that's even possible. Maybe it is. I don't know.
I don't know. It sounds like if you get it wrong, Oliver, we might have to earth you against a street lamp.
Well, either you'll get transfected or you'll get vaporized. One of the two.
It's not very reassuring, really, is it? I'm sure it's extremely small amounts of voltages involved, isn't it? It's very, very low, I would think.
Well, I think it's fairly high, just very short duration, very small pulse, very, very, very short.
Oh no! See, I didn't look at the voltages in question. Well, I would have thought it would have been an extremely sort of low amount of voltage.
250 volts.
Yeah.
Yeah.
Well, that's the same voltage as our utility sockets in the UK here. So
yeah,
240. So it's even higher than ours.
Yeah, except it's only on very briefly. Like we're talking about, like I think milliseconds or microseconds. So if we have it on too long, then you will light up like a light bulb.
Don't worry. I was always taught it's not the volts that kill you; it's the amps.
Yeah, that's true. That's true.
Yeah, it's the ampage, isn't it? But yeah, I'm sure it's perfectly fine. But yeah, I honestly think it's probably got more sort of application for sort of ex vivo. Due to the logistics. But if you could overcome the issue or the challenge of distributing it, as you say, it could be useful.
Right, because one of the issues we talk about with senolytics and senolytics not so much if they're small molecules, but if they have to be large molecules delivered to cells, then it's the issue of how do you deliver it to a whole bunch of different heterogeneous tissues and organs and cells scattered all over the place, right? That's really what we're talking about here. If you're talking about, whole organismal rejuvenation, right? Otherwise, we're just dealing with localized tissues. We're dealing with like organ by organ basis and that sort of thing. So if we really want to, take a giant leap into the future, we're talking about modifying everything, right? So that's still science fiction at the moment.
Well, science.
But we could speculate.
Yeah, of course. And science, fact often follows science fiction. I mean,
Sure.
look at the amount of things, right? And I know it's cliche to say this, but look at the amount of things in Star Trek, for example. I'm talking about the original series in particular. Mm-hmm. Next Generation, maybe. Mm-hmm. But if you actually look at the show and you look at a lot of the technologies in it, and you go, hang on, we've already got that. And it's like, so sometimes science fiction isn't always that far-fetched. I mean, certainly, isn't there like, the medical tricorder in Star Trek? I recall recently seeing a challenge, and someone's issued a challenge and a prize to create a real-life functional tricorder, medical tricorder. And I'm thinking, are we in Star Trek? But, it's crazy because a lot of the technologies that Gene Roddenberry was writing about back in the 60s and 70s, and now they're all starting to arrive, and it's like crazy.
Just get rid of the color red from your wardrobe.
unless you're in charge of engineering. I never understood that.
Yeah.
But I never understood the uniform colours because they changed them and they didn't really make sense. Why is Scotty, an engineer, got a red shirt which was supposed to be security? It doesn't make any sense. And but it really doesn't. So if any of our audience watching actually knows the answer to that, we'd appreciate finding out what is the deal with the Star Trek uniforms.
Yeah, I think the general answer would be that they were a show on a very tight budget, so a lot of stuff had to be recycled.
But yeah, it's it's definitely true about the red shirts. They always pretty much die.
Yeah.
I sometimes wonder what would happen if red shirts had a battle with stormtroopers, because the stormtroopers can't shoot and the red shirts die anyway. So
I think it would be a battle that never ended.
it would probably be a never-ending battle. Yeah.
Yes.
If you had the rebels, they were awful. They just die anyway. So I swear in some of those films that the rebels just die. They just fall down, and I'm like, no one even shot that guy. I don't know. It's crazy, but yeah, there's nothing wrong with a bit of speculation, a bit of fun.
Yeah. So anyway, delivery methods. That's something, that's another big chunk of what needs to be, pushed forward as far as technology is concerned. Not just the means to change the cells, but also, the way to do it. And all of these things all these technologies interlock, they go hand in hand, right? If you need to figure out how to reprogram cells, but then you need to figure out how to get the stuff to the right cells, right? So all of this stuff is necessary, technologically to get it all to work. All the pieces have to fit together. So progress on many fronts.
Yeah, well, one of the biggest challenges is with gene therapies in particular is actually getting it in the payload itself. So, I understand they've had success with a new type of CRISPR, for example. Was it CPF1? I'm guessing without actually checking that. New type of CRISPR, it's got a better affinity with mammalian cells. And they were saying because it packs smaller, they can get more of it into a viral vector. So they're able to do multiple gene edits all at once, which is good news, isn't it? Because the Cas9 is pretty big.
Yeah. Speaking of Cas9, we just had some bummer news. We were working on a technology here at the lab, and we got scooped. The paper was published. So it was basically fusing a recombinase protein, Rec-A, to Cas9 to basically increase localized homologous recombination, so increase the efficiency of gene targeting. So we were setting up experiments here doing just that. And the paper came out we missed it, but it came out in April. And guess what? It works.
You basically got beat to the punch. Yeah.
Yeah. But on the bright side, we our idea was sound.
And the other upside as well, of course, is you've then got replication. So you've repeated what they've done, and vice versa. Had they done it?
Well,
so
we didn't finish the experiments, so we were still building them. So we still have a couple other tricks up our sleeves, some other methodologies, but that was a real bummer.
Yeah, I can imagine. But I suppose to take the long view, as long as someone gets there and gets the results, I suppose that's all that really counts at the end of the day. It's like I say with aging. I don't care whether Sens gets it right or whether Glasgow gets it right or whether George Church gets it right. As long as, A, people are out there and they're testing these things and getting the work done, and B, one of those people gets it done. I mean, as long as we win the race, right?
Well, sure, yeah. That's great. Great outlook to have. Great outlook to have. When you're not the one being scooped.
Yeah, I understand. It is. It is. I suppose disappointing. But I suppose I do take the long view and those sorts of things. So
But yeah. Anyway,
it happens. So anyway, I mean, next month, we're going to be talking about a rather interesting subject, aren't we?
Oh yeah, it's going to be rather broad and theoretical, and covers a lot of ground.
Yeah, we're going to be discussing the very important hallmarks of aging, which is, in my view, an excellent model of how and why aging happens.
Mm-hmm.
It's also a damage repair approach, very similar to SENS. In fact, I'm going to go out on a limb here and say I think that paper was very much inspired by SENS. They are very, very similar, but we can obviously delve into that next time.
Yeah.
Should be interesting. Yeah,
I'm sure.
Yeah,
like Aubrey, maybe. I'm sure he might have some choice words.
So yeah, that would be good. Yeah, well, we can ask him, but let's So yeah, I'm looking forward to that one, and yeah, we'll get that done. So yeah, I don't know if you anything to add or anything else.
One other question. So I'm blanking on the journal clubs we did in the past, but did we cover a journal club that had Campisi as an author, and this was the senolytic in vivo senolytic delivery that was targeting, I think P53 BCL2 complex. You remember that, Steve? Or did I write a review paper on it? I can't remember for the blog.
BCL2 is the anti-apoptosis pathway.
Yeah.
I've written about it, and I think you've written about it.
But we didn't do a journal club article on it, no?
I don't think we have, no.
The reason I mention that is that paper, well, we have a... so that paper, it was mentioned to me by our new SAB member, Robert Rice from University of Arkansas, and he would love to basically do a co-journal club with us and discuss the paper. And I told him that I told him it was very familiar, and I can't remember if I did a journal club article on it or I just wrote a blog article about it. But I think that was a blog article. And if that's the case, then I think that would be really cool.
Congratulations! I agree. That's wild.
Thanks.
Yeah.
What do you think, Steve?
Now we need to just tap into all the universities at once, right?
We seem to have lost camera feed briefly. It does seem that the audio's coming through, but the camera's not. So we've got some shenanigans. Yeah. But the audio is definitely coming through. I've just checked, and other people are telling me that the audio is coming through. For some reason, we seem to have lost the camera. We're just trying to recover that.
Well, maybe it's a good time to kind of sign off because I think we've covered everything. Maybe we should. Yeah, maybe we should somehow put together a compilation of like any harebrained ideas we get from reading these papers, like at the end. It's like, have you ever read Nature? There was a column I used to love called Daedalus, where I forgot the author, but he basically made these really wild speculations on hypothetical technologies that were based on reality, and some of his predictions actually came true. Sort of like Arthur C. Clarke predicting, the need for geosynchronous orbits for satellites, for communications purposes, and that was way back in the 40s. I don't know. It'd be kind of interesting if we could make such kind of technical predictions, like the whole electroporating face mask thing.
Yes, that would be a good idea for a show. And get some beers in and speculate. It could be like that drunken history show. That would be right.
Exactly, drunken biotech.
Okay, so we're having a bit of a meltdown now with the display driver. So yeah, okay.
We should sign off.
It's just going to explode very shortly. I don't know what's gone on there.
Joel, I think we're going to sign off. We're having technical problems at this moment.
Sounds perfect timing.
All right, man.
I love this.
It's a display driver issue. So thanks for joining us, Joel, and hopefully you'll join us again. And yeah, just to let you know that Joel is one of our lifespan heroes, and thanks to his support and the support of all our heroes, we're able to bring you these informative science shows. which is great. And yeah, so thanks for everybody for joining us. And if you'd like to check out the Lifespan Heroes, if you go to lifespan.io/heroes, you can learn more about how to become a hero yourself. Right. Thanks very much for joining us, and we'll see you next time. Hopefully, without any more technical issues.
Thank you.
See you all next month.
You.
Thanks, everybody.
Thanks, Joel.